university or another research position?
It's a research institute.
Oh, cool.
Thank you.
And when would you be like physically making the trip? Or is it going to be remote for a while?
No. I'll be moving about 3-4 weeks from now.
Wow. Well, best of luck in the new, well, not a new place for you, but the new position.
Thank you, much appreciated. Do you want me to share my slides or the screen, or do you want me to wait?
We can wait another minute maybe, and then.
Oh, I see, because you're actually sharing this slide.
Yeah, Tracy has an intro slide, and then
okay.
I'll start introducing you, and then she'll stop sharing, and you can start sharing your screen if that works.
I see.
All right, I think we got a pretty good group going, so I think we can get started. Welcome, everyone. Very happy to introduce our CGD seminar speaker this week is Dr. Precious Mongwe, who we all know here from NCAR. Dr. Mongwe is a postdoctoral fellow in the oceanography section of CGD. However, I just learned that next month, he will be starting a new researcher position at the Council for Scientific and Industrial Research, which is based at the Center for High Performance Computation in Cape Town, South Africa. So congratulations, Precious, on the new position. We'll certainly miss you. He received his bachelor's in chemistry from the University of Pretoria in 2011, and his master's and PhD in oceanography from the University of Cape Town in 2018. He then joined NCAR as a postdoc in November 2018. He has participated in multiple oceanographic cruises, including summer and winter cruises to Antarctica. He's also received multiple awards at international conferences, including Best Junior Oral Presentation at the Council for Scientific and Industrial Research, NRE Science Week in 2016. His research interests include the ocean carbon cycle and the impacts of climate change on ocean biogeochemistry and ecosystems. Today, he will speak about climatic controls on metabolic constraints in the ocean. Precious, the floor is yours.
Thank you. Thank you very much. Let me share my screen. Can you see? Can you see my screen?
Looks good.
All right. Okay, thank you. Thank you very much. So before I proceed with the presentation, I wanted to also take a few minutes to really express my sincere gratitude for the opportunity that I've had to work here and to work with some of you. It was really indeed a fruitful time. I learned quite a lot while I was here, and as well as enjoyed the interactions that we've had with some of you. And as I move on, I hope that we'll continue with those engagements concerning work as well as outside of work. So I'll be continuing to use CSM, so I'll be taking CSM with me, and I hope that's another natural attractor for us to meet as our path going forward. So thank you. So today I am talking about climatic controls on metabolic constraints in the ocean. And so this essentially will give a good perspective of the work that I've been doing here at NCAR and during my postdoc, and as well as some idea of a fraction of some of the work I'll be doing going forward. So this was funded under the Deoxygenation project, which was funding my PhD, and here in the title you see the PIs on the project, PIs of this project, as well as collaborators in the work that I'll be presenting today. So as a way of starting, I'll start by, let me see if I can move. So as a way of introduction, I'll start by saying oxygen is essential to organisms. And this is somewhat of a common sense or a no-brainer, as you put it in this nation. And but the reason why this was a slide is because we are talking about the ocean medium. We're talking about the ocean medium. So, and in the ocean, oxygen concentration is not uniform. So it varies in space and as well as in time. And so the implications of that is that not everybody can live everywhere. So it determines, so these gradients then determines who lives where and how far can who travels. And so to illustrate this is actually the top figure, the top panel in this image. So in the x-axis you have oxygen concentration, and in the y-axis in the top panel you have ocean volume. So the blue line it shows the global ocean. So essentially this is a PDF of oxygen. So you see that indeed there is a variation of oxygen. There is spatial gradients in oxygen volume in the ocean, and the implications of data as well is is particularly that organisms cannot live everywhere, and this is shown by the bottom panel here. So x-axis you have oxygen concentration in micromoles again, and then you have species, and the y-axis you have fraction of species. So essentially this is fraction of species that are habitable under this range of oxygen concentration. So for example, if we take fish, which is shown in orange, if you start from the subaquatic conditions, you see that almost no fish can survive under those conditions. But as you increase oxygen concentration going to the right, you see by the time that you get to this hypoxic threshold, which is about 60 micromoles, you can accommodate about half the fish. And so as the oxygen increases further and further, you see that by the time you get to about 30 micromoles, you can accommodate almost all the fish in the ocean. So essentially this forms the basis of why oxygen is essential, and as well as in the context of marine ecosystem with the perspective that I'm looking at today. And so, but to give a brief explanation or a description of the mechanisms that sustain these gradients in oxygen, I have this cartoon here which describes the physical and the biological mechanism of oxygen in the interior ocean. So if we start from the low latitudes, so low latitudes are here, I hope you can see my mouse. So the low latitudes are generally warm. So because they are warm, so solubility is poor in low latitudes essentially. So the ocean is giving out oxygen, so you are degassing. But also you have a shallow mixed layer depth. So you have shallower mixed layer depth. And between low latitude as well as high latitudes, you have primary production or photosynthesis, which is an energy production process. So essentially you consume CO2 with nutrients and then you produce oxygen. And so that organic matter that's formed, when it sinks below the MLD, go deep into the ocean, it essentially breaks down at depth at the reverse process of respiration, where oxygen is consumed at depth. So this process at depth consumes oxygen when to break this organic matter, producing CO2 and nutrients. Yeah. But in higher latitudes, so when you look at higher latitudes are generally cooler. So you have higher solubility. So your solubility is high. So but as well you also have deeper mixed layer depth. And because of you have high solubility, you have high mixed layer depth. So that then creates a ventilation pathway essentially. So then you're actually ventilating the deep ocean through high latitude by you supplying oxygenated water to depth. So I think, so this is a brief overview, and there are details here, but I think this will be sufficient for my presentation today. So the reason why this consensus, or the perspective in which I'm looking at today, I'm looking in the context of ocean deoxygenation. And to illustrate this idea, and deoxygenation in the context of climate change or global warming. And to illustrate this, I have this figure here. So in the x-axis, you have temperature change. Y-axis, you have oxygen change from 1970 to 2004. And so in the different colors, the different colors here, these lines represent different models, so different CMAQ-5 models. And the black line and gray, the black line, which has gray shading, is the CSM large ensemble, which is what I'll be using for most of the presentation going forward. And in yellow here, or golden, depending on the eye spectrum, it is the observations, all right? So what we see, generally, we see this negative correlation that as the ocean warms, there is a decline of oxygen that we see. And by the way, this is the ocean top kilometer. And we see that in actual fact, the rate in which of oxygen decline with warming is much faster in the observation with respect to the model. And actually, this particular idea of why the models underestimate oxygen, it's a whole different talk and partly still not least understood. But it's not something that I'll be talking about going further. So I'll be here in this particular slide. The ideas that I want to articulate is that the negative correlation that we see, that the negative correlation between warming and oxygen change. But more so that because we do, in the previous slides, I introduced this idea that when the ocean is warmer, you have poor solubility. So we do expect that when the ocean gets warmer, you have poor solubility, which is illustrated by this cartoon here, that the cooler temperatures you have high solubility. So we do expect that. But when we plot the effect of solubility, which is when we only plot the direct effect of temperature, is shown by this dotted line here, right? So what we see that actually the models and as well as observations show the oxygenation, the rate of deoxygenation that is much faster than the temperature component alone. So that means that there are changes in biological processes and as well as in circulation that are playing a role into this deoxygenation that we are seeing. So it's not only the solubility that is playing a role here. All right. So now, what's particularly concerning is that the ocean is projected to keep losing oxygen as it gets warmer, and this is shown by the figure, and this is illustrated first by the figure on the left here. This is so temperature change and oxygen change the y-axis. So this figure is similar to the previous slide, but it's extended to the end of the century. So what I want to highlight here is that going further, we see that indeed there is a deoxygenation that we see with warming, and it's generally greater than the solubility component alone. So this is based on CMAQ-5 models. And here on the left, I show the CMAQ-6 model and CMAQ-6 models, and this time in the x-axis we have time, and the y-axis you have oxygen change. So we see that indeed there is the oxygenation that we expect in the future with climate change. And but the reason why this is important, or it's a concern for marine ecosystems, is that we do know that metabolic rates increase with temperature for ectotherms. So ectotherms are organisms that don't regulate their own temperature. So they rely on the environmental temperature to regulate their own internal temperatures. So this is shown by this cartoon here. So the x-axis you have temperature, y-axis you have metabolic rates and locomotion rates. So knowing that metabolism is an energy consumption and energy demanding process. So you require oxygen to produce ATP essentially. So we see that as temperature increase, you have a higher metabolic demand. However, in the previous slides, I showed that there is this negative correlation that we see. So already by this, you can see that you are going to, at some point, you are going to run into stress, organism stress. You're going to stress some organisms because you're warming the ocean while decreasing oxygen. So essentially, this forms the basis of the problem that I'm addressing in this study, or this study focuses on, to say, so we see that we're losing oxygen as the ocean is getting warmer, but we know that organisms requires more oxygen at higher temperatures. So essentially the question is, where are these impacts happening, and how and when can we detect them, and which organisms are most impacted? Because as we have seen that in actual fact, the concentration of oxygen varies in space and as well as in time. So even if oxygen decreases the same amount everywhere, the effects are still going to vary in space. But also we do know that organisms have different sensitivities to oxygen and temperature. So that's as well a spin into the analysis. So this study, I'm trying to address those questions of where are these changes happening, where can we detect them, and what are the species that are most impacted. But going further, I want to introduce another spin in the analysis, which is the idea of natural variability. So usually when we think about climate change, we think about it in the context of ecosystems as this idea of losing breath, right? That you're going to increase, the ocean is going to warm until the organisms are not able to survive or are wiped out. So you're going to, the ocean is going to acidify until you lose fitness. But when we look at nature, it's arranged in a way that natural variability is an important component of this. So to frame my analysis within the context of natural variability, I have this figure here just to illustrate the approach. So this figure is based on the large ensemble, and it's a work that is based, that is from the NCAR. So this is just temperatures in the US, right, from the large ensemble. So starting from the left, the top panel is the ensemble mean, so the average of all members, and the middle panel is the warmest ensemble, and the bottom panel is the coolest ensemble. And now in the middle panel here, you have a time series from the large ensemble as well as observation. So the black line is the observation record from about 1910 to about 2010 or so. And then the blue lines are the warmest ensemble, and the red line is, so the blue line is the coolest ensembles, and the red is the warmest ensemble. So for example, if you take the city of Mazatlan, right, in Mexico, and you look at the variation, you look at the natural variability in the black line, you see that, you have a variation that's about 1.5 degrees maximum. But if you compare that with another space and time, you take Seattle, for example, which was selected here, you see that actual natural variance there, it goes beyond 2 degrees in some points, right? So natural variance varies in space. And so in the context of ecosystem, what this means is it means that an ecosystem an organism that lives in Seattle, it can survive under this condition. It can survive under 2 degrees variance in oxygen, in temperature. So it means that in actual fact, even if they are stressed by these temperature anomalies, but they have not reached a tipping point, so they can still regroup under these anomalies. But when you introduce the climate change component, which is now, I want to illustrate using the panel on the left, is that what you're doing is that you're moving the regime. So this is, if you're moving the regime to actually a very warming region. So you're taking everything up. So even if the amount of natural variance doesn't change, the organisms must be much closer to their physiological threshold, that even the 2 degrees or the 1.5 degrees anomalies might be much more impactful to ecosystem, or might be much more harmful. And in actual fact, when you take a global average, you don't see much of that component. So you see that natural variability actually is very low when you take a global mean, and you don't almost see a difference between the largest and the warmest ensemble, which is shown by the bottom panel there. So this is this then introduces another spin to say, what is the impact of climate change within the context of natural variability? All right, so now that we've introduced that, so I can introduce the tools, and so I'll be using the metabolic theory to interrogate these questions that I've just raised. And this slide just gives a brief overview of the framework of the metabolic theory which I use. So to briefly go through this equation, so on the left you have the two top equations there represent respiratory efficacy, which is essentially the supply of oxygen into an organism. And so this is a function of the gas transfer between the body and the water, as well as body mass and PO2, so the partial pressure of oxygen. And the second equation there just shows the equation that represents the gas transfer. So essentially it's an Arrhenius function that have a temperature dependence, and you have the Boltzmann constant that deals with the gas diffusion. And then, so the middle equation, so then the third equation is the metabolic demand. So that's a function of the metabolic rate, body mass, as well as you have a temperature dependence, and you have which deals with the sort of the kinetics of metabolics in terms of the molecular reaction, and as well you have the Boltzmann constant dealing with the gas diffusion. So now, if you divide this supply by demand, essentially you can simplify life a little bit. So you can have phi, which is actually a work of one of the original work of one of the PIs of the project. So you can have phi, which is the metabolic index. And essentially what that means is that when phi is above 1, it means that you have enough oxygen to metabolize at the resting state, essentially. But when phi is below 1, you're not able to sustain metabolism for resting state. And so if you simplify this division, you have this equation here, which is like the division of the two top equations. But an additional component that I add in this analysis is that, or that we add, is that we divide phi, because this is based on the resting state, we divide phi by phi crit. So phi crit is essentially the minimum metabolism that an organism requires to sustain an active state. And so that can be determined experimentally. So if you divide by phi on both sides, so then you have phi crit. And what that does is just that now when phi is above 1, it means that the organism can sustain an active state. And when it's below 1, it's not able to sustain an active state. So this is actually meaning that phi above 1 means phi is above phi crit. So I put prime here to differentiate between the resting state and as well the active state. And all of this is simplified by this figure on the right. And the x-axis you have time, sorry, in the y-axis you have temperature, and the y-axis you have PO2. And so somewhere in the middle of that, you have like a species tolerance curve, essentially, which is like, so essentially PO2 at phi crit. And above that curve, you have phi prime above 1, meaning things are habitable, and phi below 1 means it's unhabitable. So in this particular case, I have to say that an organism might still survive, but it's not able to reproduce, move, or anything like that. So it's not contributing to the welfare of the organisms. So this is the approach that I use. So now to give this a bit of a physical meaning, I take the temperature dependence E0, as well as AC, which is the hypoxic tolerance, to illustrate much more the sort of physical meaning of this in the next slide. So now in the x-axis, you have in the left panel, in the x-axis, you have temperature sensitivity, which you've seen from the previous slides. In the y-axis, you have the ecological hypoxic tolerance. So this is the sensitivity of metabolism to oxygen. So the temperature sensitivity essentially just mean how much the metabolic demand of oxygen increases with temperature. So when E0 is high, it means that there is an exponential increase. There is a much faster increase in the oxygen demand with temperature. When it is low, there is sort of a slower increase. And the hypoxic tolerance means when it's high, it means that the organisms have a higher, is able to metabolize at very low oxygen concentration. But when it's low, it means that it requires a lot of oxygen to metabolize. And I've shown a few species here to illustrate which species lives, which species is where in this spectrum. So this is based on the observations, about 61 species from observations. Now in the right panel, I illustrate, so I sample this E0 and AC at 5 different places to illustrate what it looks like, all right? So I sample low E0, low AC, which is in blue, essentially the 10th percentile. I sample the median E0, median AC at 50th percentile, and as well 90th percentile at green. And you have in purple, you have a high E0, high AC, low E0, and a high AC, low E0. So this is shown by the colors in the right panel. So the panel, they correspond to the percentage averages on the left. So if we start on the blue line, which is low E0, low AC, so you have this bell curve shape, and essentially it means that so when it's too cold, essentially you have gas transfer becoming so difficult because of it's getting too cold. Essentially, you demand a lot more oxygen, even though the metabolic demand of oxygen is lower because of it's too cold. Essentially, is to help with the gas transfer. And as you increase the temperature, you get to some medium, to the sweet spot, And as well as when you go to higher temperatures, now you have a higher metabolic demand, so you see an increase in the metabolic demand for oxygen. So this plot sits up high in the oxygen concentration, which means that remember you have low AC, so and as well it sits very, very high there. So essentially in the y-axis, this is actually PO2 at 5 Cret. So above this curve you can sustain a metabolism, and below it you cannot. So if we move from blue to purple, so keep A0 AC and increase AC, you just move this curve down here. So now you see that you have a lower, so you can metabolize even at lower oxygen concentration. If we move from blue to red, so keep AC constant, you increase A0, we see that we come with this red curve, which just shows that there is an exponential increase in metabolism as you increase temperature. So the disadvantage of a species like that is that by the time that you get to 20 degrees, it's very hard to metabolize because the oxygen that you demand is just too much. And when you take the green, you have high E0, high AC. So you have the green curve, which can metabolize at even lower oxygen concentration, but you can metabolize, even organisms can survive even at higher temperatures. So as you will see later, that this is actually this is an organism that can live almost anywhere in the ocean. And in orange here, you have the median species, which is shown in the dotted line here. And I put in the dotted line because of going forward, I'll be using this median species, which is shows the most abundant species in the ocean to illustrate in the analysis if I don't mention which species I use. So you see that this background here shows the density or the frequency of occurrence. So this is the most abundant species. So now to look at what this looks like in the ocean, I sample now the transect in the North Pacific. So the plot here is the plot on the left is the plot you've seen in the previous slides. In the x-axis here, at the bottom here, you have PO2 and as well as temperature for a transect in the North Pacific, and is shown by this transect here. And so now, so this grid essentially is this, it represents that, and I've marked the colors to that correspond to that. So now if you start with the blue, which is the blue curve at the top here, where low E0, low AC. So this, I have plotted the metabolic index here. So when it's below 1, you cannot survive. When it's above 1, you can survive. So when it's clear, when the color is clear, it means it's below 1, so you don't have any species surviving. So if we start by this one, we see that actually this organism prefers higher latitudes, but it avoids much colder regions, which are close to the poles, right? Close to the shelf. It's too cold to survive in both latitudes, but so they prefer somewhere like in the subantarctic zone in the Southern Ocean, as well as in the 40s in the northern hemisphere. And but as you go to the right, now you're sort of increasing your temperature sensitivity. So you are bringing this curve downward. So the advantage of this is that now you can metabolize it even at higher and higher temperatures, as well as at lower oxygen temperatures. So you see that when you move from blue to red, you essentially follow the temperature contours that you can metabolize at higher temperatures as well as lower concentration, but you're still avoiding the tropics because of it's too hot, so they can't live there. And if you move from the blue up to the purple, you see you are just essentially moving down this curve. So you are decreasing the demand for the threshold for metabolism. So you can live at most parts of the ocean following the oxygen contours. And the green shows high E0, high AC. So you can see that this can live in most parts of the ocean, including OMZs, which are very low in oxygen. So now that's the framework. So to show this in another way that is perhaps might be more relatable, I introduced this idea of the oxygen safety margin. All right. So this is just the difference between the local oxygen or the local PO2 versus PO2 at 5 Cret. And it's illustrated by this figure here. So this is the figure that we have seen in the previous slides, but this time I've colored red, meaning uninhabitable or unable to sustain an active state, and green, meaning able to sustain an active state. So if you take some idealized state of the ocean, which is shown in this marker here, X, the difference between that state to the critical oxygen is the oxygen safety margin. So it means that when you cross this line, if you cross this line, you're not able to survive. And so that distance is oxygen safety margin. And in x-axis, you can do the same. You can find the x-intercepts, which tells you the temperature safety margin. So you can think about it like a safety tank, more or less. So, and this pattern is shown here, which is the same grid. So essentially here, when it's brown, it means you're not able to sustain an active state. And when it's blue or green, you are able to sustain an active state. So if you look at this pattern, it's actually much similar to the previous slides, except that now it's showing the brown showing that you don't have enough oxygen to sustain an active state, or have green shows that you can metabolize. So this you can read there. So now that we have built that up, so now we can actually look at how things look like when we take all the species. So now on the left here, I have this panel showing temperature and PO2. And so the different lines here just sort of idealized, representing different organisms of different sensitivities to oxygen and temperature. So it's like a species tolerance curve for different species. So if you take those and take them through the metabolic index framework using the large ensemble, temperature and oxygen field, you arrive at a figure like this, which is at the bottom here, just shows the number of viable ecotypes. So a relatable term here is the oxygen, is the species richness. So this represents something like that. So you see that if you take the upper ocean, for example, you see that in the tropics, you see you have lower species, you have lower amount of species can survive there. So about the 10%, about 10 ecotypes out of the 60 can survive. And as well, you have the highest richness at the subarctic zone. And this in the upper ocean, this is primarily driven by temperature, because the oxygen is generally abundant in most parts of the upper ocean because of you have efficient AC fluxes. So oxygen is pinned to the atmospheric CO2, whereas temperature have a much more, varies much more zonally. So this is the pattern you see, and when you go to 200 meters, you start seeing the importance of oxygen. So these areas of low richness, these are areas of OMZs, which are very low oxygen concentration. And you sort of have a similar pattern where you have a higher occupation in the subarctic zone, and as well as avoiding the very high latitude because of it's too cold. And 500 meters shows a similar pattern that we can infer from the temperature field, that we see here. So this is how things look like, how the things change in the future. So now, I just described here this slide, I'm just showing how I define my natural climate, right? So this is a time series that I pick 200 meters from the large ensemble to illustrate the point. So you have temperature at the top, PO2 at the bottom, and this is time in the x-axis, all right? So the large ensemble started 1920, and but somewhere from about 1965, we start seeing a trend in oxygen, in deoxygenation, and then somewhat of a trend in temperature. So then I define my natural climate as 1920 to 1965. But I have to say that just to word of caution that I define, so this is my natural climate. The natural climate means before the effect of climate change, essentially. But there are cases where I differentiate in terms of the present climate, which is today. So how does things change with respect to today? I call that my present climate in this analysis. And this is 2005 to 2020. Right, so now let us look at the changes. So now starting with the net changes with respect to the natural climate for oxygen and temperature. So now the top panel, most of you are familiar with it, just shows the that actually the ocean warms nearly everywhere. And essentially you see the largest warming in the upper ocean. And as you go to depth, you see lesser and lesser warming. So it warms nearly everywhere with few exceptions like the warming hole that we see here in the North Atlantic. So the ocean warmed the largest the surface in the upper ocean because anomalies propagate from the upper ocean. Whereas when you look at oxygen in the upper ocean, we see very little, very little changes except for areas like the Arctic Ocean, which is partly due to the melting of ice. And this is essentially the same idea that you have efficient AC fluxes. So essentially the upper ocean is pinned to atmospheric oxygen. But when you go to depth, you start seeing a larger spatial gradients, right? You see that larger changes in space. For example, in the Pacific Ocean, we see the largest losses, large losses in oxygen, but there is some gains that we see in the Atlantic and as well as the Indian Ocean. And at 500 meters, you see some changes. You see that there's a spatial heterogeneity. So now to put these changes into context, I look at the relative changes between oxygen and temperature. So I represent this as a regression between PO2 and temperature. And the reason why this is important is that in the context of the metabolic index, it is important, it's important what oxygen does when oxygen changes. So the higher level emergent pattern is a negative correlation, which we have seen from the beginning, but at local scales, there is some variability that we see. Now, so to illustrate this, the top panel here shows the natural climate, which is 1920 to 1965. And the bottom panel is the first trend, so 2020 to the end of the century. So at the upper ocean, you generally see, in the natural climate, you see a weak positive correlation, except for parts of the Atlantic Ocean. But as you go to, when you go to 200 meters, you start seeing a positive correlation in the lower latitudes, and high latitudes, you see a negative correlation. So what this means is that, so red is good. So red means that as you increase temperature, you are supplying the oxygen demand that is required. So essentially, you are delaying the impact that you have into metabolism. But blue is bad because of you are decreasing oxygen. You are warming while you are decreasing oxygen. All right. So, but then when you go to 500 meters, you see that there's an inverse of what we see in the 200 meters, where you see negative correlation in the lower latitudes and positive correlation at high latitudes. If you go to the first trend, the upper ocean is much similar to it's mostly similar to the natural climate, except for the Arctic Ocean. And then, but when you go to 200 meters, you start seeing some significant differences between the natural climate and the first trend. For example, the Pacific Ocean, you see that there are some areas close to the California current that was positive correlation in the present, so it's negative correlation in the future. So this means that things go bad in the future, essentially, because not only are you losing oxygen, but you're seeing a stronger negative correlation. And when you look at 500 meters, you see that actually the pattern that we see in the natural climate is actually opposite to what we see in the first trend. So this gives us an idea of where we expect the largest changes. In some places, things move from blue to red, so it means that things get better. And these are regions like the Western Pacific or the Indian Ocean, part of the Indian Ocean. So now let us look at the metabolic index. So I have like sort of three ways to look at the implications of these changes. All right. And as a start, we start by the metabolic index for the median ecotype that I've shown in the beginning. So this, the panel here that I've shown is it's respect to the natural climate. All right. So generally we see a decrease in the metabolic index in most parts of the ocean, except in the upper ocean, except for the high latitudes. And as you'll see, and this is because of warming here, the organisms escape the cold bias, the cold intolerance. So things get better in some parts of the high latitudes, mostly the Southern Ocean, because of organisms now can accommodate more species, as I will illustrate actually in the next slide. But generally, we see a loss of metabolic index everywhere, which is what we expect. All right, you're increasing, you're warming the ocean, while decreasing oxygen, so you're decreasing the metabolic index. And this is shown by the figures. If you look at these figures of the net changes that I've shown 2 slides ago, it corresponds with this slides, that areas where we see gain in the metabolic index are areas that we see oxygen gain in the future. And here in this panel here, which is shown by this mesh, is I just difference with respect to the natural climate, which I need in the next few slides. So I just want to illustrate that if you difference with respect to the present climate, essentially we see the same pattern as we see with respect to the natural climate, or more or less the same. So now let us look at, so we see that there's a decrease in the metabolic index. What does this mean, essentially? So to try to do that, the first way we can look at that is to use this idea of the safety margins, which I've shown, and I've illustrated with using oxygen, but here I show both oxygen and temperature. So this is the similar figure that we have seen from a few slides ago in the introduction. And now I've just annotated, I just put some labels in it. So the blue line, which is represented by the marker that I've shown. So this is something like the present climate. And going forward, we expect a decrease in oxygen while it's getting warmer, which is a general pattern. So there's this negative correlation. So climate change is represented by something like this orange, by this gray line, essentially. So what it's doing is that it's moving the local habitats towards the species, towards this critical point, which is shown by the thick gray line. So where the ocean crosses this curve, or where habitats crosses this curve, they are not habitable. And now, so to illustrate that, here I take the top kilometer and I find sort of the local adapted species. And so you can do this using sort of you can use the frequency of occurrence essentially to find an average locally adapted species. So I take that for the top kilometer and I take an average of the top kilometer. And this is the figure that we are seeing in the middle panel here. So this is the present line, all right? So the blue means that you are further away from the threshold, means that life is good. And the clear lines, which are white, shows that you're actually much closer to the physiological threshold. This is for the temperature safety margin at the top, and at the bottom is the oxygen safety margin. And if you move from the present into the future, now, and actually the letters that you see here is just to help you correspond with the figure on the left, but you don't need it. So now when you look at the future, we see that we move from clear to red. So essentially areas that are much closer to the physiological threshold, they crosses to the red. So it means we see that in temperature and as well as in the oxygen safety margin. So it means that actually climate change displaces locally adapted species. So it has an impact. So it's encroaching in the habitat of locally adapted species. Now, so this is just based on the local adapted species. Now let us look at what things look like when we consider the entire species collection. All right, so this figure shows the number of viable ecotype. So by the way, I realized that I didn't mention what ecotypes are, and most of you are much familiar with species. So ecotypes just means a group of species that are adapted to the same conditions. So it's a relatable context to species or species richness. So in this figure, what I show here in the x-axis is temperature, y-axis you have PO2, oxygen, and projected into the figure is now the number of viable ecotypes, which is shown by the color bar. So essentially what this is that it just tells you what species that are habitable under these conditions of temperature and oxygen for the range of species that we have according to the observations. So the emergent pattern here, it's something like at very cold temperatures, we see that you have low habitability, which means, so this shows you the cold intolerance. So it's too cold, gas diffusion is too hard. So the only few species can survive. But as you warm, you get to some sweet spots here somewhere about 15 degrees, 10 to 15, where you have a sweet spot where most organisms are happy under those conditions. And as you warm further, you lose more habitat. So you are not able to accommodate more species at higher temperatures. So essentially then, so when you get to higher temperatures and lower oxygen, you accommodate more species. Now projected into this figure in the ellipses that you're seeing. So this is now a PDF, essentially it's a joint distribution of oxygen temperature for the ensemble mean based on monthly data sets, all right? So now I sample a few regions in the ocean to illustrate the impact of climate change. I'm just doing this for the upper ocean because I want to make a specific point that it's not everywhere where you are losing habitat. There are regions where you are gaining habitat. And this is illustrated, for example, if you take the Arctic Ocean here, we see that we see a general gain in oxygen over time, as you've seen from the previous slide. So you move from the blue is the present, the red is the future. So essentially, you see that there's an alignment of the ellipse, and as well it moves higher up. So it moves towards more species, more fitness for a lot of species. So you accommodate more species in the future because of your moving up there and with a slight warming. And if you take a region like the upper ocean, the Southern Ocean, in the upper ocean, the present ellipse is blue, red is the future. So things move to the right, essentially. So you are warming the ocean because you are warming. So as you move to the right, you see that actually the species in the Southern Ocean, it escaped the cold bias. So there are species that are not able to survive because it's too cold, but in the future, they'll be able to survive because of you are warming the ocean to try to the sweet spot. So the Southern Ocean is moving toward the sweet spot, at least in the upper ocean. This is just a sample at 50 meters. So, but if you look at the Pacific Ocean, so if you look by the Northern Pacific, for example, shown by this large ellipse, you see that we are moving from a darker color towards lighter in the color bar here. So essentially we are losing species because of this is because of the warm intolerance, all right? It's too warm, so you are losing species. Whereas in the Southern Ocean, you are gaining more habitats because of you are escaping the cold intolerance. Here you are losing habitats because of warm intolerance. And in the upper ocean, we see that you're just essentially moving to the right. And what that means is that you are just losing appetite because it's just too warm in the central tropics. Another component that we see from this, which is an interesting effect, is that this can tell you something also about extreme events, all right? That if you have warming extreme events in the Southern Ocean in the future, they will be less harmful than in the present because of you are much sitting at the sweet spot, whereas extreme events in the North Atlantic as well as the Central Pacific, they will be much more impactful because of you're actually even further up into lower species, all right? So the ellipse, for example, in the North Atlantic, in the North Pacific, it crosses more species habitat in the contours here. So now as I draw towards the end, I want to pass by this point here to make a point that just to have some slide that shows some optimism, essentially. So we see that, so when we compare, so by the way, the analysis that I've been, what I've been using so far is the RSP 8.5 scenario, which I think I showed in the previous slides. So if we compare the RSP 8.5, which is the business as usual scenario with respect to the Paris Agreement, the 1.5 scenario, we see a dramatic difference. And so this is shown by the panels on the left here. So the top one is the 50 meters, the bottom one is 500 meters. Now, if you take, for example, the Central Pacific, if you look at the lower latitude, it shows in black 20 south to 20 north. We see that in RSP 8.5, we lose nearly half the species, all right, in the RSP 8.5. So this is, the x-axis is the ecotype fraction. So this is similar to the very first slide I showed the species fraction. So this is the number of ecotypes that are habitable under this region. So when it's 1 is 100%, above 1 you are more than 100%. When it's low 1, that's where you're losing species, is the difference. And this difference with respect to the present climate. So you see that as you go in RSP 8.5, we lose about nearly half the species. And when you look at the 1.5 species, when you look at the 1.5 scenario, we see that we lose only about 10% of the species. And so we see that actual fact that the 1.5 really makes a big difference, essentially in this. And an example, another example we see at 500 meters, where we see that we lose about half the species in 1.5 and we lose only about 15%. And this is shown an average of the top kilometer on the right here, where we see that the difference is actually quite dramatic. So it shows that actually if we are able to push the 1.5 scenario, if we stick to the Paris Agreement, there is some, it can have a big implication into the marine ecosystems. Now, so as I conclude, I want to come back to this idea of natural variability, which I introduced earlier. So now what you're looking at here, so this is natural variance. So this is the 1920 to 1965. So this is standard deviation of temperature, PO2, and as well as the metabolic index for the 3 depths that I've been using. So we see that if you look at the metabolic index, we see, starting with temperature, we see that you see a larger standard natural variance in the upper ocean that decreases with depth. Whereas for oxygen, you still see some higher natural variance at depth, right, with respect to the upper ocean. And when you look at the metabolic index, we see a natural variance that is much closer to the temperature in the upper ocean. And at depth, we start to see more similarity with depth. But when you look at the depth, we see that the metabolic index natural variance is much more similar to what we see with respect to the natural climate. Sorry, there is some noise that is coming outside. I don't know what's happening, but I'm almost at the end. So this is what we see. So the question is, so how much is the climate change impact with respect to this natural variability? And when you do that, so I use the time of emergence to look at that question. So what we see here, what I show here is in the x-axis you have time, y-axis you have temperature, PO2, the metabolic index for the median species. This is my second slide. So in the gray lines is all the ensemble members, and the black line you see the ensemble mean. The purple, the green is the 2.2 2.3 percentile and 97 percentile. So the time of emergence is defined by the time when the ensemble mean crosses the two percentile standard deviation. So we see that, so what the important factor that I want to show here is that we see that because of in this particular grid point, there is an increase in temperature while oxygen is decreasing. The time of emergence for the metabolic index is actually earlier than both of them. For example, if you look at the temperature emerges around 1950, PO2 emerges about 1940, and the metabolic index emerges somewhere about 1930. And this is because of the warming, is because of warming, there is a decrease in oxygen as the ocean warms. So essentially this accelerates the rate of climate change. That's why you see the earlier emergence in metabolic index. If they were both going up, metabolic index will probably emerges earlier because we are delaying the impact into ecosystem. And this is my final slide, which shows the time of emergence for the entire space, for the top 3 depth, for the temperature, PO2, and as well as the metabolic index. So in the upper ocean, we see that temperature has already emerged, and it emerged later and later in depth. PO2 doesn't emerge in the upper ocean because we have very little changes, and we see more emergence with depth. And we look at the metabolic index for the average species, it says that it might have, the climate change signal, we might already see it in most parts of the ocean. And as you go to depth, we see later and later emergence. So I think I'll stop there, and I'll leave the summary up there, and I think I've addressed most of these points that are here, and I'll be happy to take questions. I apologize for the noise. There seem to be some construction outside, which I didn't anticipate, but they are going to start early today. Thank you.
Thank you so much, Precious, for that really interesting talk. Really nice to see the ecological impacts studied here. See some virtual applause, and the noise isn't too bad on my end, so don't worry too much about that. So we have about 10 minutes for questions. If you have a question, you can type it in the chat, or you can use the Zoom raise hand feature to ask a question out loud. And that button is under the reactions at the bottom of the Zoom screen. So any questions for Precious? I do have a question to start us off. I found the time of emergence results really interesting to compare the ecological metrics like the metabolic index, emerging so much earlier than some of the biophysical metrics. I guess my question is, how might we account for ecological adaptation in the future projections, if that's something you've thought about, or if that's something you think might impact the results, especially when we look at like a high emissions scenario?
Yeah, no, I think that's a very good question. So the idea is that in this analysis, we don't address the idea of adaptation, but the simple answer is that we don't know, right? So we don't know whether these species are going to be wiped out, or are they going to, essentially we don't know whether how fast is the ability for the species to adapt with respect to the rate of climate change. So if they adapt faster than the rate of climate change, probably they might survive, but that remains actually mostly unknown. So I don't account for it in this study. I think that's the simple answer.
Great, thanks. Other questions? Joni, go ahead.
That was an excellent talk, Precious. I really enjoyed it. I think you covered a lot of interesting questions. I might have missed in the beginning. You talked about ectotherms versus endotherms, and I suspect you were only looking at the ectotherms in this process, but can you say anything? I mean, you also had some examples of some of the species, examples of where they fell on the temperature versus metabolic demand curve. And do you have any generalities about which types of species would be most vulnerable to this in terms of bottom dwellers, phytoplankton, across the realm of what you were looking at? Can you make some generalizations about that?
Yeah, that's a good question. So I mean, the first point is that I start from zooplankton up, essentially. I don't consider phytoplankton going down. But the emergent pattern, what I can say is that at the upper ocean, the stress is mostly temperature because of it getting warm. But at depth, it's much more the oxygen component, that's playing a role. So you are eliminating, you are losing organisms due to loss of oxygen at depth, what upper ocean is due to temperature. But so the species, so if you look at the trait space, so essentially organisms that are more sensitive to oxygen at depth, you are losing a lot of those organisms. Whereas in the upper ocean, these organisms that are more sensitive to temperature that we are losing. So that's the general statement that I can make so far. But I cannot comment on endotherms because of I didn't consider them here. But yeah, that's a good question. Thank you.
Thank you.
Other questions? Dave, go ahead.
Yeah, thanks, Precious. I'm just wondering what the impact of any sort of biases in the model might be. I've heard from, don't know the details, but I've heard from Keith and Matt and the other Keith Moore about, issues with oxygen minimum zones and in CSM1, I think, and can't remember if they've been resolved in CSM2. So, can you comment on that, the impact of biases?
Yeah, thanks, Dave. Yeah, that's a good point. Yeah, so I removed this slide here, which I thought I was going to run out of time. But so in CSM1, what we see is that, so generally the model compares well to observations. But the point that we are raising about OMZs is a valid one, because the only thing that we see is that at regions of low oxygen, so the OMZs, which is the bottom is CSM and observations at the top, World Ocean Atlas data, we see that OMZs are particularly extensive in the model, all right, when you compare them to the observations. And the implications of that is that the model is quite moderate in representing the metabolic index. So you see that, for example, the observation says, well, the Atlantic Ocean is not quite zero yet, but the model already says that it's actually zero, nothing lives there. So the general idea is that the model is modest, with I think that which is a good thing, which it says that the model is telling you the least impact. If our scenarios on the basis, if our scenarios is right, this is the modest scenario essentially. Does that answer your question, Dave?
Yeah, thank you. Yeah.
Maybe this is related, but you showed the figure at the beginning where the relationship between oxygen and temperature in the observations was a lot different from in the models. Do you have anything more that you wanted to add on that point, or is that like a subject of future work?
Yeah, no, I think it's subject of future work, but I think what you're asking is why is the correlation changing between the observations why is the correlation changing between the neutral climate and the future, the climate change? Is that what you're asking? This slide?
Yes, I'm thinking about at the very beginning when you showed the relationship, the trend in oxygen versus temperature and how it was steeper for the observations.
Oh, right.
Models. Just that overall.
No. I wouldn't. I think it's a complicated. I don't think there's a simpler answer in terms of why the model does is underestimate. There is some ideas about why that is, but it's not an area that I've worked a lot in. So to avoid misleading you, I won't comment. We can have a much more discussion about that offline. Yeah.
Okay. Any last questions for Precious? All right, I'm not seeing anything else. So thanks very much, Precious. Next week, we have Kasia Tokarska from ETH Zurich, who's going to be giving the seminar. So hope to see you then. And thanks again, Precious, for a great talk.
Appreciate it.